So, looks like the number of attendees is climbing. Shall we begin? My name is Dr. Berkey, Dr. Adam Berkey. I'm a neurologist and a member of the NANS Education Committee, as are all of the others who will be speaking tonight. This talk is on basic mechanisms of spinal cord stimulation, both what is known and about the action based on experimental studies, as well as some of the theory for how spinal cord stimulation may work, and some ideas that could be the subject of future research. As a neurologist, I did basic neuroscience research in anatomy and in neuropathic pain. So for that reason, I'm going to start tonight's presentation by discussing the substrates of neuropathic pain in the nervous system, starting with the primary afferents and the dorsal horn of the spinal cord. So I'm advancing the slide now. There we go. So that's us. I'm the guy on the left. I practice in Washington State, Puget Sound area in private practice. So primary afferents in the dorsal horn. As most of us know from our first year of medical school, the pain and temperature fibers are considered to be the small unmyelinated C fibers, as well as some of these small myelinated A delta fibers, which have a particular pattern of connectivity within the dorsal horn. The nociceptors, which sense pain, temperature, as well as itch, are classified anatomically into peptidergic and non-peptidergic afferents. One of these peptides clearly being substance P, as well as CGRP and TRPV1. Both types of fibers are glutamatergic, which is the main excitatory neurotransmitter in the nervous system. The peptidergic neurons are receptive to painful heat, and they terminate in lamina 1, which is the most superficial portion of the dorsal horn, and the outer portion of lamina 2. So in our diagram on the right, you can see Rexed's lamina, and you can see the superficial dorsal horn, which is the term for lamina 1 and lamina 2, is there on the right, where you see cell bodies with synapses with A delta and C fibers. And then you see lamina 5, which is in the neck of the dorsal horn. And that receives afferents from mechanical A delta fibers, as well as some non-nociceptive afferents, which are the large, more heavily myelinated A beta fibers. Anatomically, the non-peptidergic fibers project to the inner lamina 2. They tend to receive more mechanical painful stimuli, and they can be, in research terms, described using the lectin IB4 antigen by immunohistochemistry. Next slide. So this shows you in a little more detail how the connections are made. The peptidergic and non-peptidergic A delta and C fiber nociceptors, along with low threshold A beta mechanoreceptive afferents and low threshold C mechanoreceptive afferents, are all sort of pictured there along the top in different colors. The superficial laminae projection neurons are exclusively in lamina 1. So if you see the green neuron there, all the way on the left at the very top, that's a projection neuron, and it receives afferent input and is subject to processing by the other interneurons there to the right. And its output is it decussates and travels in the anterolateral system up to the forebrain in the spinothalamic tract. So the projection neurons are in lamina 1, and the processing is occurring in the deeper lamina 2. There are wide dynamic range neurons which respond to pain as well as other non-nociceptive stimuli in the deeper lamina 3, 4, and 5. The inputs from the non-peptidergic afferents convey their inputs to the lamina 1 projection neurons via a polysynaptic excitatory pathway that include what are called central and vertical neurons. So you can see in red there's a vertical neuron which has these extensive dendritic processes, and there's a central neuron there, both of which are excitatory. Their output is modulated by inhibitory interneurons, which are tonically active, that contain GABA and glycine in normal conditions. And those are under the control often of non-nociceptive afferents. And it's this type of circuitry which gives the basis for the gate control theory, whereby non-nociceptive sensory input can reduce nociceptive input transmission to the forebrain through the connectivity in the dorsal horn. So I show this picture just visually to make the point that the output from lamina 1 is modulated by a columnar organization of excitatory and inhibitory interneurons, and that these columns extend through lamina 1 and 2, but they stop at lamina 3. So this is a image of a rat that had Pseudorabies virus injected into the parabrachial nucleus. So the parabrachial complex is near the brachium pontus, the nuclei which transmit nociceptive signals. Pseudorabies virus was injected into that nucleus, and then Pseudorabies virus will travel from one neuron to the next in a pathway on the order of about 1 neuron in a chain every 24 hours. And normally the rat can survive 4 or 5 days. And so you can get tracing of pathways in the central nervous system by infecting a nucleus or a site with Pseudorabies virus and then sacrificing the animal after several days and seeing which neurons the Pseudorabies virus has managed to infect. And you can see nicely there in the dorsal horn that we have a columnar arrangement of nociceptive projection neurons in lamina 1 with the interneurons that control them in through lamina 2. There is an influence of the large heavily myelinated proprioceptors and light touch fibers. These are the fibers that travel in the dorsal columns and synapse eventually in the gracile and cuneate nucleus. The projection neurons are in the neck of the dorsal horn. The wide dynamic range neurons that they control are in the neck of the dorsal horn. You can see that these A beta fibers, they have axon collaterals that will control and excite inhibition and control inhibition both on the projection neurons in the neck as well as other excitatory interneurons, and therefore they have the ability to reduce nociceptive output when functioning correctly as they project rostrally. So the basic anatomy, the fundamental anatomy in the healthy individual is that there are small fiber afferents, which are classified as either peptidergic or non-peptidergic. These peptidergic and non-peptidergic afferents have a different population of neurotransmitters. They have a different pattern of synapses, whether in lamina 1 or the outer or inner lamina 2. They affect processing in these columnar organizations of excitatory and inhibitory interneurons, as well as they can synapse on the long dendritic processes of nociceptive projection neurons from lamina 5. The system is influenced by large fiber input onto inhibitory neurons, interneurons, and onto projection neurons in lamina 5. And lamina 5 has the wide dynamic range neurons, which are responsive often to mechanical nociceptive stimuli, mechanoreceptive, whereas temperature is in the superficial dorsal horn. So that's normal sensory processing in the dorsal horn. But under conditions of nerve injury or other types of chronic pain, there's a change to what goes on in the dorsal horn. And one thing is that cells die. And so this is an image of a silver stain. They're showing after nerve injury, they're showing cell bodies that are lost, and you see there's a concentration of them up in the superficial dorsal horn as well as throughout the spinal cord after axotomy. Most of us probably know that there are two types of cell death. There's excitotoxic cell death, which leads to necrosis from calcium overload generally, where there is then an autophagy and an inflammatory response to the cell death, and the cell products are eventually eliminated. And then there's apoptotic cell death, which is programmed cell death, and this is where the signal is made to the cell and relayed to the nucleus, whereby the cell will, in a very organized fashion, commit suicide and simply become pycnotic and disappear without any inflammation or response by the body. This is also in response to glutamate at times and calcium. There's the activation of caspases, lipid peroxidation, and gene activation with chromosome condensation in the nucleus that leads eventually to apoptosis and a characteristic type of DNA splicing into fixed length of DNA repeats that you can determine on a blot that apoptosis is going on. And it is known that apoptosis and cell death occurs in the dorsal horn with nerve injury and neuropathic pain models. In fact, it also occurs in response to morphine. So intrathecal morphine has been shown to induce cell death in the dorsal horn. And cell death seems to occur more among the GABAergic and glycinergic interneurons than it does in other populations or interneuronal populations of the dorsal horn. And this may be because it's those inhibitory interneurons which are under tonic excitatory control. And so if there's a burst of excitation with nerve injury, they're the ones who receive the brunt, more glutamate than even the projection neurons or whatnot that are directly excited by the primary afferents. This loss of inhibition in the dorsal horn leads to phenomena like itch, spontaneous itch, as well as allodynia, and is thought to contribute to hyperalgesia and allodynia in both human and rat models. By the way, the article that some of these images are taken from is listed there as Benaroch dorsal horn circuitry. It's a very nice article that's basic science, but published in a clinical journal. If anyone wants a copy of that paper, I think it's probably the single best review for these topics. I can give a copy to Lily at NANS, and maybe we can give people copies of that paper if they want to get a hold of it. But loss of GABAergic interneurons is clearly a mechanism after nerve injury of hyperalgesia and allodynia, and a reason why neuropathic pain is difficult to treat, because the native inhibition is lost. Another thing that happens with nerve injury is deafferentation. So if there's a nerve injury or if there's a toxic metabolic process like diabetes that's affecting the peripheral nerves, you will have a loss of small C fibers, which can be detected by skin biopsy. The nerve densities go down in sites affected by the neuropathy, and the remaining small fibers become neuritic and swollen and unhealthy. And the loss of these small C fibers leaves a void in the superficial dorsal horn, and nature abhors a vacuum. So what you can see is that non-nociceptive afferents will grow from the underlying lamina 3 and take over and make connections with nociceptive neurons, interneurons, and projection neurons in the superficial dorsal horn. So the image on the right, there's control, the control level in A and B, where you can see that there's no staining for the for the mechanical non-nociceptive fibers in the superficial dorsal horn. It's restricted to deeper levels of the dorsal horn until you deafferent the level of that dorsal horn. And after nerve injury and deafferentation, nerve fibers which are not nociceptive will grow into nociceptive areas of the dorsal horn. And this is another reason for allodynia and abnormal pain processing that's difficult to manage in patients with nerve injury. Another mechanism of neuropathic pain with nerve injury is activation of microglia. So microglia, well, first of all, glial cells make up over 70% of the total cells in the central nervous system, and they are classified as astrocytes, oligodendrocytes, and microglia. Microglia are the resident macrophages of the CNS. They come from the primitive yolk sac. It's not known what exactly activates the microglia. It hasn't been determined, but there are several candidates that for transmitters that activate the microglia, including a substance called monocyte chemoattractant protein 1, and there's a metalloproteinase 9, whose expressions are increased in dorsal ganglion neurons after peripheral nerve injury and may play a role in activating the microglia. But as you can see in a spinal cord to the left, if you look for MAP kinases and other signs of microglial activation, on the side of nerve injury, you can see an upregulation and activation of these cells. And the pathway in my doing research for this is shown on the right seems to be the best established, which is ATP active on purinergic receptors, which then release BDNF. BDNF, it's the track B receptors on neurons and will ultimately lead to depolarization and activation of neurons transmitting rostrally pain signal. And this area of research is the newest, and it's the one that's been growing the most in the last 5 to 10 years. A lot of the things that I've been discussing with regard to GABAergic interneurons and loss of cells and aberrant connections has been around for a number of years, but the microglial research is something that is received a lot more attention recently, and there are both drugs under development as well as spinal cord stimulation strategies that are meant to turn down the activity of the microglia. So I wanted to point out at this time that although it's difficult sometimes to understand what particular pathologic changes may be going on in a particular patient, we should all be aware that empirically, there is this concept of sensory profiles, and it's something that's done over in Germany as a matter of course in sort of large state databases, looking at the characteristics of the neuropathic pain, which are felt to relate to underlying structural factors like we've been discussing, such that we can use some Bayesian concepts in prior probability maybe in the future to select patients with certain sensory profiles that may have a certain underlying pathology for certain therapies. And so for instance, the group, Ralph Barron, the senior author on this article, has published the most on this. They've described looking at thousands of postherpetic neuralgia patients that using a factor analysis that essentially there's 3 types of postherpetic neuralgia. So with or without numbness, with or without allodynia, with or without cold allodynia or heat allodynia, with or without hyperalgesia, with or without electrical stimulation. Looking at all of the characteristics, the clinical characteristics of the neuropathic pain quality and evoked characteristics of the neuropathic pain. Using a factor analysis, they've determined that there's maybe 3 kinds of postherpetic neuralgia. And whether that correlates with the success of certain therapies or not is one direction that clinical research is going, or with medication, selecting patients with certain neuropathic pain characteristics that may or may not respond to gabapentin. because if it's a neuropathic pain condition that lacks the GABAergic cells to work on, perhaps those are the patients that gabapentin is ineffective and so forth. So there is a clinical correlation for all of this basic science. So the three plasticity items I've brought up are the loss of the GABAergic inhibition, the formation of new synaptic connections, and the activation of microglia that are releasing excitatory substances that activate nociceptive signaling. So the nociceptive pathways in the dorsal horn have to actually go somewhere. We have to be conscious of pain for us to report the experience of pain. The nociceptive projections from the dorsal horn terminate starting in the brainstem, in the nucleus of the solitary tract, the medial brainstem reticular formation, the caudal ventral lateral medulla, the lateral parabrachial nucleus. That's a big lamina 1 relay, which is where the Pseudorabies virus was injected in the rat model. The midbrain paraductal gray receives input, the hypothalamus receives input, as well as the thalamus, and two nuclei in the thalamus in particular. One is the ventrocaudal medial-dorsal thalamus, and the other is a nucleus known as the ventromedial posterior nucleus. So we're going to talk about those specifically. Now, the medial-dorsal nucleus defines the projection territory of the medial-dorsal nucleus, defines the prefrontal cortex. The medial-dorsal ventrocaudal subnucleus of the medial-dorsal complex projects to the anterior cingulate cortex, area 24, or specifically area 24C in the cerebral cortex. And then the ventromedial posterior nucleus, which is part of the ventroposterior medial complex of nuclei classically. That nucleus was defined back in the 90s by a cluster of calbindin reactive small C fiber aprons as being a distinct nucleus. And it was studied in the context of this model called the Thunderbird grill illusion, which is simply what you see there. It's this device that will stimulate a part of the body simultaneously with non-painful cool and non-painful warm sensation. And when you do that, a person will report that they feel a burning pain. So that although you're not providing a nociceptive input, the confusion created in the nervous system by a simultaneous cool and warm stimulus results in an experience of pain which is not associated with tissue damage. So it's really a peripherally evoked type of a central pain, and it was described in the 19th century by a guy named Thorsten Thunberg. And the anatomical basis for this illusion of pain was found out through basic research and functional imaging studies to be a relationship between a certain population of C fiber that projects through the ventral posterior thalamus to the insula, and then another set of C fibers that projects through the medial-dorsal ventrocaudal to the anterior cingulate. And this was functional imaging of 5 different conditions: painful heat, which is 47 degrees centigrade, painful cold, which is 5 degrees centigrade, non-painful cool, non-painful warm at 20 and 40 degrees, and then the grill illusion, which is the combination of the 20 and 40 degree temperatures simultaneously. And when you do functional imaging of this phenomenon, what you see is that the insula is activated under all temperature conditions, including the grill, whereas only those conditions where the patient reports pain do you see activation in the anterior cingulate cortex. So what this implies is that the so-called lateral pathway of lamina 1 to the insula through the parabrachial nucleus and through the ventromedial posterior thalamus, that's a site of sensory integration for a range of sensory inputs, and it integrates it as well with cardiovascular and visceral responses and emotion. And the insula you can think of as sort of the area that assesses incoming stimuli from the body, and if they fall outside a certain parameter, it disinhibits or allows activation of the anterior cingulate, which creates the consciousness of pain. And so the medial pathway, the medial pain pathway, is through the small fiber pathway through the medial thalamus, the medial dorsal thalamus to the cingulate. And the lateral pathway is a different set of C fibers with different physiologic characteristics that projects to the parabrachial and ventromedial posterior nuclei to the insula. And the insula is more evaluative, whereas the anterior cingulate is more specific to the experience of pain itself. So one can imagine that if one had a therapy that could boost the input to the insula to keep the anterior cingulate inhibited, that could be an effective pain therapy. The insula and the anterior cingulate cortex, of course, they just receive the C fiber pain and temperature input directly, but of course, once it's in the forebrain, there's a whole network of sites that get activated, including somatosensory cortex and prefrontal cortex, amygdala. And then what goes up must come down. And it's been known since the 70s that the brainstem in particular but the brainstem under the control of the forebrain, it's also clear, can send a signal down to the spinal cord at the level of the dorsal horn to either inhibit or enhance painful input. So the descending modulation can be either facilitatory or inhibitory. The periaqueductal gray is one known site, but it acts through the rostroventral medial nucleus of the medulla, so the RVM there. That's the site that contains serotonergic neurons that project to the dorsal horn and modulates nociceptive activity. And then the other big projection is from the locus coeruleus. And the locus coeruleus projects to the dorsal horn with noradrenaline. And these are the two main transmitters that are involved in descending modulation to the spinal cord and dorsal horn. And therapies which can increase descending inhibitory controls are also analgesic. So it's not always a matter of just blocking the transmission rostrally of nociceptive input, but if you can increase descending inhibitory controls, you can also get a reduction in pain. This is my final slide, and it shows an SCS there positioned over the dorsal columns, and that can produce activation, presumably that increases inhibition in the dorsal horn, and the columnar network in the superficial dorsal horn maybe decreases activity. It's possible that spinal cord stimulation ascends rostrally and either boosts input to the insula, which increase descending inhibitory controls, or via another mechanism. The main transmitters that we think about with descending inhibition are shown there as noradrenaline and serotonin. There are endogenous opioids in the dorsal horn as well, particularly dynorphin, as well as enkephalins on the primary afferents. And then there's a host of other interleukins, CGRP, peptides, GABA, and cannabinoids. These neurotransmitter systems are all under the influence of depolarization and action potentials, which are the mechanism of action of spinal cord stimulation in all its various forms. So this is my last slide, and I wanted to leave it there and maybe move on to our next speaker. If there are questions, I think what we'll do is, unless there's a number of them, I think what we'd like to do is maybe hold on to them, and we'll answer them as a group at the end, and there may be questions that kind of get answered in the course of subsequent talks. So unless there's a ton of questions, we'll probably just continue with the speaking and handle questions at the end. Okay. Dr. Anatescu?
Yep, I'm here. Cool.
Okay, take it away.
I will. Thank you very much. So we'll talk briefly about those three mechanisms of action, those three waveforms today. My name is Magda Anatescu. I work at the University of Chicago. And without further ado, let's see if I can move it. Moving.
Yeah, there's a little delay.
Yep. Well, oh, there you go. Sorry. Yes, it started moving. Those are my conflict of interests. And then this is my first slide. So we talk a little bit about the neuromodulation of the spine, what specifically we target with our neuromodulation. Some sort of a type of waveforms we know, and we guess and study the mechanism of action, and a little bit about implications to the patients. In terms of basic mechanisms of action, the way the spinal cord stimulation works, or suggested working, is basically activation of the dorsal column axons. Once you apply electrical current into the epidural space on the dorsal columns, you have a transmission and initiation of an action potential, which goes orthodromic away from the soma and antidromic towards the soma. It comes with a high degree of selectivity because the large diameter fibers that do have a lower threshold for firing and being activated. Our understanding of the mechanisms of the spinal cord stimulation comes from the finite element models and of stimulation. And the basic understanding is that we actually activate the large fibers, but much less so cell bodies, and also the dorsal roots are activated usually much easier than the dorsal columns. The presence of the CSF is also very important. If you have very little CSF, the threshold of activation is much less, as opposed to higher amount of CSF, which actually shunts the influx away from the stimulation. So therefore, that is probably why you have much less stimulation in the cervical area than the mid-thoracic and lumbar area. Moving on to the stimulation targets, the characteristics of the targets are, as we know from the gate theory, or classically, the neurons. After an electrical stimulation, you create an electrical field, which does change the transmembrane potential, action potential, generation of an action potential in axon, and this action potential is transmitted, as we mentioned, antidromic and orthodromic. We have also the stimulation threshold, the amount of current which is required for depolarization, and then also it depends on the characteristic of the axon. So if you have a very thick, big axon, like in A beta fibers, you have a thicker myelin sheath and you have many Ranvier nodes, and therefore the electrical impulse just like jumps in between those, and then it activates usually a little bit quicker. The classical activation, the classical mechanisms, as I mentioned, is neuron-centric, but they are currently we are currently discussing more mechanism of action with multiple targets. And this is basically what we know about the analgesic effects of the neuromodulation. As I mentioned, the classical conventional paresthesia present spinal cord stimulation has been very well studied. So we know their effects on the neurotransmitters, on synaptic depressions, wide dynamic neuron, glial cells, supraspinal, and axonal conduction. However, the newer waveforms like high frequency burst are a little bit less understood, and their mechanism of action are emerging. Let's talk a little bit about the high frequency stimulation. This is a new waveform, new in like 6 or 7 years, not very new, but still new in the terms of how old the spinal cord stimulation, the neuromodulation is, more than 50 years. In terms of the definition, is a paresthesia-free neuromodulation technique. It does offer a program with a 10,000 kilohertz with a low amplitude and a very short duration, but with pulse wave of 30 milliseconds. What is characteristic about it, and very clear when you actually do have neuromodulation, neuromonitoring applied in those patients, you have complete blackness into the neuromonitoring records. So there is no waveform on the neuromonitoring, which is in alignment with the absence of a sustained action potential in the dorsal columns. The proposed mechanism of action was based on animal and computational model, and is actually activation of a wide dynamic range neuron by modulating those neurons with dorsal horn fiber recruitment. There is some controversy about this as well. Those mechanisms of action were described based on in vitro and ex vivo experiments on animals, primarily adult male sprague dolly rats. And basically what it was found and suggested is that the mechanism is by a low intensity subsensory threshold at the frequency of 10 kilohertz may activate the inhibitory interneurons without activating the excitatory ones. There are also some effects on neurochemical changing for glutamate and MAP kinases. Those papers are emerging. They're, as you can see, very, very new, and they're trying to justify and understand the clinical, the mechanism of action of a clinical finding. Moving on to the next target, which is microglia. We heard a little bit about their role into the the amount and the role into the spinal cord. They're everywhere. As Dr. Berkey said, they're very, very numerous. 70% of the cells into the CNS is basically microglia. They're involved in modulation of the neural networks, calcium hemostasis. They do orient the neurons, the laminar orientation of those, and they're also involved in vascularization and myelination. So activation of microglia, it is known that it does produce chronic pain. So the question arises, if we actually activate if we actually apply electrical current to those cells, what do we actually do? Do we modulate the pain? Do we decrease the pain, or we don't have any effect? So, as mentioned, our understanding of the classical mechanism of action for spinal cord stimulation being neurocentric, with the axonal activation of paresthesia perception and neurotransmitter release, the electrical stimulation can also cause the glial cell depolarization and the glutamate release. So they do respond to those electrical impulses as well. In order to understand how do they respond, there has been a lot of research and a lot of activity recently about transcriptonics and proteomics. What it is, these are the sciences to study the effects of electrical stimulation on gene and protein expressions, basically correlating the expression profiles with the biological process, thus understanding how the electrical signals can modulate the biological processes themselves, primarily the glial cells. Several early papers 6-7 years ago showed that there has been some effects by applying the spinal cord stimulator to rats into the genes expression for those sacrificed animals. And basically the main effect is on the gene expression associated with immune and inflammatory responses. The specific waveform used to understand and explain this activation of the glial cells is called differential therapy multiplex, and it was primarily designed to modulate the neuron-glial interaction towards a naive state, basically normalizing the immune system while creating neuroprotection. Several papers did suggest that this is the case. By looking at the low rate, high rate DTM and naive patients and studying the microglial transcriptons, it was suggested that by applying the DTM, the state of the cells was going towards the resting state, the naive state, which is pretty much very evident in this 2020 paper and this table, which showed that if you can see here in this column, in this line, when you apply this specific waveform, you can recover from the injury between 79 and 85% of all of the elements, all of the anatomical presence in the spinal cord: microglia, astrocytes, oligodendrocytes, and neurons. Oops, sorry. And there are also animal models. Sorry. Moving on to the evoked compound potential, action potential, Ecap, which is an advantage for what is the advantage of having this waveform and this possibility to our toolbox. What it allows us to do is that to provide basically improved pain relief with in real-time feedback and closed loop systems, and correlating very closely to paresthesia. by recruiting the A beta fibers in the dorsal columns. Animal models that did suggest that there is a possibility to create this in real-time conduction measurements for the Ecaps. And then when you actually think about the neuromodulation and activity with a medication, it makes you wonder what can we do to kind of improve our outcome for the spinal cord stimulation. And there are several papers which several new research showing that there are patients which had 2 or more classes of medications preoperatively before implantation of spinal cord stimulation, which had a lower explantation rate comparing to those with 1 or none. This is a discussion how the spinal cord stimulation is correlating with the medication. It is a discussion, a completely new discussion for a completely new topic. But just to give you an idea, there are some medications which can be influencing our effects of spinal cord stimulation. And then why this is happening is that because there are 3 components for this elements, the neurophysiological component, pharmacological component, and psychosocial component. So when you actually do apply spinal cord stimulation, neuromodulation for those patients, you try to decrease the excitability of the dorsal horn neurons, acting basically as a membrane stabilizer. So you don't necessarily need another medication to do the same job. However, many medications can act as membrane stabilizers as well, which can be synergistic with the effects of spinal cord stimulation. The centrally acting medication as well suppress the firing of inhibitory neurons, which can contribute to the effects of neuromodulation. Pharmacological agents, though, do create and have a lot of side effects, so it is very important to kind of differentiate between the effects of the spinal cord stimulation and the medications involved. And ultimately, if you want to win a patient from the medications they are on, there is a psychosocial component that the patient has to be committed to the therapy and be willing to wean those medications off. So in conclusion, we briefly touched base on those 3 mechanisms of action. High frequency activation of the glial cells and Ecap. Those mechanisms of action are still under development. They're not completely elucidated. And then despite of the fact that this neuromodulation is old by more than 50 years, we still are learning and trying to understand what is the actual mechanism of action by which those patients which we implant the spinal cord stimulator are having so much pain relief. Keep in mind that there is a correlation between the medications we use in spinal cord stimulation, and they can be synergistic or antagonistic depending on the medications. Another mechanism which is on the way to be elucidated in the near years. Thank you very much. And now, I think Dr. Ferguson will be next. I am going to be muted and without the camera. Please take.
Hi, thank you everyone for joining me on this webinar, taking time out of your busy day to learn about mechanisms of action of spinal cord stimulation, action and theory. What I'm going to do is try to discuss the mechanisms of action like everyone else did, but what I'm also going to do is try to clinically correlate the mechanistic theory into real world practice. As far as disclosures, I have no disclosures. I'm not a consultant. No one's paying me. I'm not a shareholder in anything. So as some quick background, I know my esteemed colleagues went through this, the anatomy of the gate theory, gate control theory. So the short version of this is that you have a painful stimulus coming in, peripherally activating a nerve. That stimulus travels through the A delta C fibers, through the primary neuron, it goes to the Y dynamic range neuron, and then it travels up to the thalamus and into the brain, causing pain. Traditional gate control theory shows that if you stimulate the A beta fibers, what that does is it sends a signal to the interneurons and the Y dynamic range neurons, creating a paresthesia sensation dampening the input on the Y dynamic range neurons, which then decreases the sensation of pain in the brain and the thalamus and emotional centers. So said another way, the patient is still sensing pain, but by stimulating the A beta fibers, they're feeling paresthesia overlap, which dampens their sensation or perception of pain. That's classic, standard spinal cord stimulation theory, as discussed by all my previous speakers. Now there's something called the surround inhibition, and that is a different mechanism than gate control theory. The person that came up with surround inhibition is Dr. Hillman and Wall. So the gate control theory, as to recollect, the gate control theory was Melzack and Wall, and that was in 1965. Surround inhibition was performed by Hillman and Wall, and that was in 1969. So the evolution of spinal cord stimulation theory and understanding from a mechanistic standpoint is between 1965 and 1970, there was the first paresthesia publications by Shelley and Mortimer, and then the gate control theory by Melzack and Wall. And that's the one that everyone's familiar with, and that's the one that is in many of the, if not all of the pain textbooks. The Melzack and Wall theory was done with an intrathecal lead. It was a single contact. What this evolved into between the 1970s and 2010 was conventional paresthesia mapping. That was between 40 and 60 hertz. It was a single source, bipolar targeting. The positive applications of this was that it was very fast onset, it was high efficiency, and medium to rapid target search, meaning you could move the lead around and find where that pain source was in the dorsal columns and try to overlay the paresthesia mapping with the pain source. This then evolved into more advanced paresthesia mapping between 40 and 60 hertz using multiple leads, different targeting algorithms between 2013 and the present. Again, above this line is the just to orient everyone, so above this line is the paresthesia world. So again, as the paresthesia mapping improved, the frequency was between 40 and 60 hertz. Again, the advantages was fast onset, it did not use a lot of energy, rapid target searching because you would just move the leads around to find where that paresthesia overlay was, allowing for pain control. And this is all using the standard gate control theory. And again, the hypothesis is the dorsal columns are being targeted. Underneath this line is subperception pain relief. So subperception meaning the patients are not feeling anything, there's no paresthesia. So this was first published by Dr. Yearwood in 2006. So there was conventional subperception threshold, meaning 10,000 kilohertz, bipolar targeting, and they started using that in 2015. The downside of subperception threshold was that it was very slow onset. It would take hours to sometimes days for this to start working, and it used a lot of energy. The recharge burden was very high, and it was very difficult to target search because you had to estimate where the most appropriate place was to place the leads to cover the area of pain, because the pain relief would take, again, hours or days. So you couldn't really do this intra-op during a spinal cord stimulator trial. This was then modified from the 10,000 kilohertz to between 500 and 1,000 kilohertz using bipolar targeting, and this came about in between 2016 and 2018. Again, this is subperception, no paresthesias. Issue was slow onset. If anyone was using the system between 2006 and 2018, they would notice that the patients would then have a trial 2 to 3 days later or a day or two later, they would start noticing the pain relief onset. This was then improved upon in 2018 to 2020 with advanced subperception, again using 200 hertz, and it was a novel field. and that was used between 2018 and 2020. Same advantages and disadvantages. It was a slow onset, high efficiency, rapid target search. So the clinical takeaway of this mechanism of action is there are two different mechanisms. There's the paresthesia, and then above this blue line, so there's paresthesia mapping, and then below that black line, there's subperception. The pros of the paresthesia mapping is it's very fast, but clinically not as effective. Subperception, it was more effective. The downside is that the clinical onset was slower. It had a higher charge burden typically, and it was a very slow onset of action. So the next iteration is surround inhibition, which is an established mechanism. The surround inhibition happened, again, way back in 1969. So to reiterate, because repetition is good, in 1965, there was the Malzakin-Wall gate control theory. Then in 1969, there was the Hillman and Wall, which incorporated surround inhibition into the gate control theory. So the surround inhibition is the next iteration of gate control theory. And at least with the pain textbooks I've read, it usually is not mentioned, but it really has been around for a very long time. From 1967 up until the present day, there's been a huge uptake in publications regarding the surround inhibition. It's gone from almost nothing in the 1960s to a few hundred, and it's almost exponentially going up in this curve. So despite surround inhibition being an established scientific literature, the question is, how does this actually impact clinical practice? So the person driving the surround inhibition theory is Professor Warren Grill at Duke University. And what he's done is he's used computational models and a lot of preclinical data that's supposed to be published out of Duke to incorporate this surround inhibition into clinical practice. This is a very busy slide. This slide was presented at NANS in 2022. The takeaway from this slide, which is very busy, is that low amplitude spinal cord stimulation, it will activate the dorsal columns asynchronously and inhibit the wide dynamic neurons. And I will go into more detail into that mechanism in the coming slides. But really the takeaway is that surround inhibition works at those wide dynamic range neurons, the second order neurons, to dampen their firing to decrease pain signaling. So I have my own podcast. It's not a monetized podcast. Basically I interview physicians that are experts in their field and try to promote them on social media. I don't get paid to do this. They don't get paid to do this. It's just a voluntary thing. So I interviewed Dr. Grill, and he goes into approximately an hour-long presentation on the mechanism of action of spinal cord stimulation, if anyone cares for a really in-depth conversation about spinal cord stimulation. And as a plug, he also talks about the future of spinal cord stimulation and what the next iteration of spinal cord stimulation will be regarding pain relief. So the new subperception spinal cord stimulators theory is paresthesia-free pain within minutes of activation. And as a side note, the reason this is clinically relevant and understanding the mechanism of action is because of how quickly it actually sets up. So I have a clinical story of, I had a medical assistant that worked within the hospital system for the last 10 years. She had horrible CRPS. It went, of course, misdiagnosed for 2 years. So no one knew what it was. They thought she had rheumatoid arthritis. They were putting her on a bunch of rheumatologic drugs. She gained a lot of weight. The CRPS became really severe. If anyone is familiar with treating complex regional pain syndrome, it can be a really debilitating, miserable disease. So she went from this vibrant, very cheerful medical assistant who worked really, really hard to really debilitated, and she couldn't leave her house. She was housebound because of the pain, because being in her car just caused severe pain. There are a lot of different targets regarding spinal cord stimulation. So What I did was I placed the leads from a clinical standpoint at the top of T11, turned on the paresthesia mapping. She got great coverage of her feet. I turned on the subperception, fast-acting subperception therapy, immediate 100% pain relief. The lady came in a wheelchair, and she literally walked out. Like, nurses were crying. It's a very dramatic thing. So while the theory of spinal cord stimulation can be dry, the clinical application is very relevant. So understanding the clinical application is very important. So the requirements for fast-acting subperception therapy is you need precise targeting, you need a proper dose, so you turn on the targeting over the area of pain, then you decrease that stimulation between 30 and 40% below perception. And typically, for those that do their own programming, it's between 210 and 230 milliseconds and 90 hertz. I will say, as a side note, from a clinical standpoint, this, in my experience, does not apply to abdominal and pelvic pain. So I have another story of a patient who had severe pelvic pain. So getting that paresthesia overlap is very difficult with abdominal and pelvic pain because you don't have that dermatomal representation. So you're just not going to get that paresthesia overlay. So just by turning on fast, you can adjust the leads to where you're getting pain relief because it's so fast. And by placing the leads for the pelvic pain at the top of T11, it was immediate 100% pain relief. The patient did well at the trial, and she continues to do very well. No opioids, and is very happy with the therapy. So basic this is standard paresthesia spinal cord stimulation theory. So you have an area of pain in this epicenter of pain. So this is the area of the spinal cord that is causing the pain. You have surrounding this pain an area of cells that cause surround inhibition. A way to think about this is if you have a poke on your hand, and I'm sure everyone is familiar with this analogy. If you've poked your hand and it hurts, if you rub around your hand, you're surrounding that area of pain with stimulation using the gate control theory and decreasing the pain sensation with that overriding paresthesia or stimulation, decreasing pain. So to orient yourself on this slide, this is the dorsal columns. These are the leads. And this is a blow up of that green is the inhibitory neurons, and that red is the wide dynamic range neurons that then will send pain up into the thalamus and into the brain, leading to pain. So as the wide dynamic range neurons or the second order neurons fire, that leads to increased pain. So as you stimulate the area of the wide dynamic range neuron, and then the area of the inhibitory neurons, there's a conflict of stimulation. So by targeting the right area, you are activating the surround inhibition. So you're targeting the areas around the pain, like you're rubbing your hand around the area of pain. That activates the inhibitory neurons, and that leads to decreased pain. However, with the activation of the pain center itself, you're also causing pain, so you're not getting a very strong amount of pain relief. So the theory would be, in real life practice, if a bee stings you in your hand and you rub around that bee sting, you'll get a fair amount of pain relief. If you start poking at where the bee stung you, then depending on how you stimulate it or rub your hand, you may actually get an increased amount of pain. And that's why there isn't a dense amount of pain relief with conventional paresthesia spinal cord stimulation. It works, but not as well as it probably could. So that's conventional gate control theory. The pro is you get very precise targeting because it's fast onset, covering the area of pain, but again, not very strong stimulation. With surround inhibition, what surround inhibition is doing through the work of Dr. Grill using the well-known, well-established surround inhibition papers from 1969, what it does is surround inhibition maximally targets the area around the pain center, activating the inhibitory neurons, which decrease the firing of this wide dynamic range neuron, and it minimally activates the wide dynamic range neuron itself. So you're maximally inhibiting it and minimally activating it. This, of course, is mechanistically influenced by the lead array. So if you have a tighter lead electrode, and if you have differing currents, you can adjust the stimulation to maximally cover the surround inhibition while minimally covering the wide dynamic range neurons. So the more options in programming, the better. So this is a study showing, the study was 41 patients, had significant pain relief from 6.5 down to 1.3. And the follow-up at 3 months and 6 months showed a decrease in pain from 8.4 to 1.6 and 1.7. And more data showing study cohort of 67, and the pain decreased from 8.5 to 2, and the pain decreased from 8.5 to 1.3 3 months out, and the typical pain relief was 0 to 2. So in conclusion, the takeaway message from this presentation is that there's the gate control theory, which is the standard one that everyone is very familiar with. And then there is the fast-acting sub-perception spinal cord stimulation pathway, which has been around for a long time. It's being worked on by Dr. Grill at Duke University. It is sub-perception, but acts quickly. So in my clinical experience, if you are trying to cover abdominal and pelvic pain that don't paresthesia map very well, you can turn on the FAST system, see where the pain relief is, and then actually modify where the lead placement is to optimally cover the area of pain. Published reports have demonstrated that fast-acting sub-perception therapy can provide effective paresthesia-free analgesia within minutes of programming. In my experience, it's probably under a minute. Thank you.
All right. My name is Iknur Talkes. I recently joined the Department of Biomedical Science at Florida Atlantic University, and I'm a canine cynomordi working on chronic biomarkers of chronic pain in SCS patients. I've been working with Dr. Julie Plissis for years, and in the last part of today's panel, I will be going over the electrophysiological markers reported in SCS patients and concepts behind the research methods. All right, this is my disclosure. Well, as we've seen in the previous talks, gate control theory explains a number of features of pain relief from SCS. However, the network proposed by this theory is insufficient to describe all of the features of the pain and SCS. And current evidence suggests that mechanism of action of these novel waveforms, such as high-frequency stimulation and burst stimulation, is complex and multifactorial, indicating both spinal segmental and supraspinal pathways. Regarding the spinal segmental pathways, we have already covered the basic concepts such as gate control theory, role of dorsal horn, large and small diameter fibers, role of microglia, tonic stimulation versus novel waveforms, computational models, major stimulation strategies, and Ecaps. And we touch base on the ascending and descending pathways. Now let's move to the brain and discuss some major concepts related to supraspinal circuits, such as pain matrix, electrophysiological testing, and cortical and subcortical signatures of pain and SCS. Well, chronic pain is a complex phenomenon with multi-dimensions. For example, studies have shown that 77% of the people with chronic pain report feeling depressed, 51% of the chronic pain patients have little to no control over their pain, and 20% of adults report sleep disturbances due to their pain at least a few nights a week. So it seems that pain perception occurs as a multi-regional activation, and broader functionality related to multisensory integration, emotion regulation, general cognitive and attention processing, self-referential processing, and other functions. Well, experimental studies in healthy controls using thermal, mechanical, chemical, and electrical stimuli, and these studies in patients with chronic and acute pain show that painful stimulation activates excessive areas of cortical and subcortical brain regions, which they referred as the pain matrix. And central pain processing is classically described in the pain matrix, consists of the sensory discriminative pathways, affective motivational pathways, cognitive evaluative pathways, and descending modulation. Let's start with the sensory discriminative pathways. Well, briefly, this circuit, also referred as the lateral pathway, includes the structures such as primary and secondary somatosensory cortices, primary motor cortex, supplementary motor area, temporal parietal junction, thalamus in the deep brain, and insular cortex up to some extent. As you may know, the insular cortex has a pretty complex function, and it describes in multiple pain networks. And these components are related to certain functions such as localization, modality, and intensity of pain, as well as the motor reflex, avoidance, escape, and autonomic responses. The affective motivational, which are shown in purple here, and the cognitive evaluative pathways shown in blue are including structures such as prefrontal cortex, insular cortex, anterior cingulate cortex, as well as amygdala, caudate, putamen, hippocampus, and nucleus accumbens. And those structures are related to the functions, pain-related functions such as suffering, anxiety, fear, depression, and cognitive control of pain. And the additional structure brain regions related to descending modulation includes periodactyl gray, locus coeruleus, and rostral ventral medulla. Well, these brainstem, midbrain, and medullary regions are involved in the descending modulation of pain. Therefore, exerting both inhibitory and facilitatory effects of spinal circuits. Well, I'm not going to go over the entire text here, but long story short, the take-home message is that pain involves coordination between these areas working together, and single anatomical areas do not drive the entire percept of pain. Now we have an idea about the cortical and subcortical brain regions involved in pain perception, but how about measuring it, right? So alternative to patient-dependent subjective outcomes, multiple objective methods have been proposed, and not only to measure the presence and level of pain, but also to characterize the mechanism of action of spinal cord stimulation. And these methods include quantitative sensory testing, blink reflex, neuroconduction studies, somatosensory evoked potentials or SSEPs, laser evoked potentials, skin biopsy, heart rate variability, pedometry and accelerometry, nociceptive flexion reflexes, and neuroimaging modalities. The neuroimaging modalities including structural imaging such as magnetic resonance imaging, MRI, or diffusion tensor imaging, DTI, functional imaging modalities including fMRI, functional near-infrared spectroscopy or FNIRS, electroencephalography, EEG, or magnetoencephalography, MEG. And finally, the molecular imaging methods include positron emission tomography, PET, and single photon emission tomography, SPECT. Well, each of these modalities has their own advantages and disadvantages, but in the next slides, we will look at the research findings from the SCS and EEG literature, which is the non-invasive, most commonly used method, which is non-invasive and the highest temporal resolution among these modalities. All right. So despite the fact that chronic pain has been estimated to affect 50 million Americans, there are a limited number of studies evaluating the neural effects of spinal cord stimulation, and only a few of them measuring the EEG in humans with spinal cord stimulation implants. The table is from a recently published 2022 review paper, which is giving us an overview of available literature on resting state EEGs in patients with chronic pain who have been treated with SCS. And these studies are all published in the last decade or so and investigating the spectral features in response to different spinal cord stimulation waveforms. Let's briefly look at the major findings in these studies. The first one from the table is a 2020 study. In this study, researchers are investigating the spectral effects of tonic, burst, and high-frequency stimulation at 1.2 kilohertz. And they are measuring the spectral, they are using high-density EEG with 61 channels. And they are looking at the EEG changes in 3 chronic pain patients with cervical SCS and 1 chronic pain patient with thoracic SCS. Because, as a side note, because cervical SCS, because of the distance between the stimulation electrode and the sensors is pretty close, it induces stimulation artifacts. So the researchers also propose a special filter for artifact attenuation in this study. So on the left-hand side, you see the average power spectra computed for each condition: tonic stimulation, which is computed relative to off state, burst stimulation relative to off, and high frequency stimulation again relative to off. And on the right hand side, it is pretty much covering the same information, but we see the subband power distribution in bar plots with statistical significance for from left to right, from tonic to high frequency stimulation. So on the left hand side, actually the major finding of this study is that all these 3 waveforms, regardless of their differences, they are inducing a reduction in the theta band power, and at the individual level, also at the population level. And they also show that tonic and high frequency stimulation is increasing high beta, these regions, high beta and low gamma power at individual and group level, and burst stimulation increasing these high beta, low gamma power only at the population level. So which means it doesn't induce the same activity in all patients. And finally, differently in tonic stimulation, it is suppressing this high gamma activity, but only at the populational level. Well, this study doesn't cover these changes related to cortical regions, but it proposes that this reduced activity in theta alpha band is consistent with the thalamocortical dysrhythmia hypothesis of pain, which is kind of indicating the, enhanced inhibition in the afferent pathway, switching to permanent burst mode, generating slow oscillations in this frequency range. The second study is from 2019. Again, in this study, researchers are looking at the spectral changes with respect to high-dose SCS and tonic stimulation. In this study, let me check that. Yes, in this study, they are including patients with failed back surgery syndrome. They are using 32-channel EEG setup. And first, they get three EEGs. One is before the SCS surgery, second EEG 1 month after the surgery, and these patients were programmed for about a month with tonic stimulation. And after tonic stimulation, they reprogrammed with high-dose SCS, and they stayed on this program for another month. On the left-hand side, you see the changes, spectral changes with respect to these 3 conditions. The pink one is corresponding to high-dose SCS, and it shows overall higher energy in the entire spectrum. When they look at the differences between these 3 conditions, they show us the highest difference located to theta or low alpha range, which is also statistically significant between these 3 conditions. In addition to these spectral changes, they also look at the functional connectivity, and among all common connections, only the beta band significantly differed in strength between these 3 conditions. And it was from, we see this information on the right-hand side, it was from left frontocentral to left temporoparietal region. And it was in favor of high-dose SCS. Well, these differences in power spectrum and connectivity between these 3 conditions lead to the hypothesis that high-dose SCS may have a higher contribution on the excitatory bottom-up pathway. And in this 2015 study, Dr. Derrida and Vanessa is investigating the spectral changes in 5 patients in burst stimulation versus tonic stimulation, and they use source localization to investigate the subcortical region activations. And they also include the sham stimulation, which is stimulation of just immediately after inducing paresthesia. Well, major findings of this study is that comparison between tonic and burst stimulation shows a significant synchronization in alpha range in the left and right dorsal anterior cingulate cortex, dorsal medial prefrontal cortex, and primary somatosensory area with burst stimulation. In terms of connectivity analysis, they revealed a significant reduction in connection between dorsal anterior cingulate cortex and right parahippocampus for burst stimulation. And overall, researchers propose that burst and tonic stimulation modulate the ascending lateral pathway and descending pain inhibitory pathway, but burst stimulation additionally modulate the medial pain pathway, which might be rebalancing between ascending pain input by medial pathway and descending pain inhibitory activity. In this 2014 study, again, researchers are looking at the spectral changes in 30 SCS patients. They are evaluating, and all these patients were programmed with tonic stimulation, and they are also comparing the spectral changes to healthy controls, 10 healthy controls. On the left-hand side, you see the average powered spectra computed for each group. The control group is shown with clear circles here at the bottom, and neuropathic pain group before SCS shown in the dark circles, and the same group 3 months SCS treatment shown in the light diamonds. The major findings are that they showed the neuropathic pain group before SCS showed a pretty strong theta band activity. When they are under SCS treatment in 3 months window, the activity in alpha range is switching from low frequencies to the high frequencies. And another finding is that the healthy control shows a pretty strong fast alpha rhythms in the spectrum. Right-hand side also showing a similar information. Here, the subband powers are compared between these healthy controls and the pain group before and after SCS. The interesting finding is that in the beta band, they showed, by the way, the stars in comparison to healthy controls, they showed that the beta band activity was significantly higher in neuropathic pain before SCS, but when the patients were treated with tonic stimulation for about 3 months, the beta band reduction showed no significant difference from healthy controls. All right, so the last study is from our recent study from 2020. So here we investigated the spatial spectral changes with respect to high frequency stimulation at 10 kilohertz and tonic stimulation. And differently, we investigated the spectral changes during the spinal cord stimulation electrode implantation surgery. So all these changes corresponding to intraoperative EEG. But consistently, we found similar results to the pain literature and the other studies. Let me walk you through the major findings that we had. On the left-hand side, you see the topographical distribution of relative theta and alpha band powers under tonic stimulation and high frequency stimulation. If you compare the top row, you don't see a significant difference between these two stimulation waveforms at the theta frequency range. But when we look at the alpha, we see an overall enhancement in high frequency stimulation, and the somatosensory area shows a significant enhancement in alpha range. When we look at the changes in the peak frequency, we also found a significant shift in theta in the prefrontal and frontal cortices in the theta band from stimulation of the 10 kilohertz high frequency stimulation. When we look at the correlation between these objective metrics that we obtained during surgery and the Oswestry Disability Index scores 3 months after the surgery, which is condition-specific outcome for the back pain, by the way, showed a positive correlation. This means patients who showed better improvement in their back pain 3 months after surgery had stronger alpha theta peak power ratio at the time of surgery. By the way, 10 kilohertz was clinically optimized settings for these patients. So in conclusion, overall tonic stimulation and the paresthesia-free stimulation waveforms may affect different pain processing pathways. Well, long story short, we can say that to better understand the network dynamics and the role of neural circuits in the perception of pain and the pain relief in response to SCS, we need better biomarkers. We need more studies exploring the electrophysiology underlying the stimulation. We need more studies showing different responses to different SCS parameters, and we need studies including well-designed experiments, which includes placebo effect. And thanks so much.
Okay. Lily, do you have any questions that have come in that you can share with us?
I have not seen any questions come in through the chat. So if anybody listening has any questions for the panelists, please type them into the chat box.
I see some. I clicked the question mark button to the left, and I see a couple, I think. So I see one that says, what is the role of S1 and S2 in pain perception? And that's related to intensity and localization, primarily. The limbic system, if the limbic system, insulin, incident, and ACE is the other ones responsible for unpleasantness. So S1 and S2, somatosensory cortex, it helps to localize pain and to encode intensity. The anterior cingulate identifies the experience as painful in some conscious way. In the insula, it's not clear. It definitely is involved in monitoring stimuli to see if they fall out certain parameters of acidosis or temperature, if it implies injury and pain, and should be flagged up as such. So that's my answer to those questions. Can anyone else see these questions with the question mark on the question mark tab there?
No, they're all being sent to you, Dr. Berkey.
Oh, okay. So it's just me. I'm the only one who can see them.
Yeah.
Okay. So there was a question, how does spinal cord stimulation explain regional paresthesia when the dorsal column fibers are arranged medial to lateral and superficial to deep? So I guess it's asking what are the parameters on physiologic activation of how do we control activation of the dorsal columns when it's obviously superficially located over the dorsal columns and the depth of the stimulation. Is there anyone that can address that maybe? The regional activation of the dorsal columns. We all know that we do it empirically when we're mapping. I'm not sure any of the research addressed that question specifically. There was a question, Dr. Telkus, about how soon do the EEG changes happen after spinal cord stimulation? Could it be used during the trial period as a marker of success?
Maybe, potentially, yes. We are also investigating that. I mean, tonic stimulation, you can immediately see the effects, right? Or the paresthesia, because of the paresthesia, but the novel waveforms, you have to wait for 24 to 48 hours to see the effect, but there is the placebo factor. So for the trial period, for about 1 week, possibly yes, but I haven't seen any studies reporting the changes in the trial period in response to stimulation.
I mean, I guess it would be interesting if we had a database of people that we trialed and we compared their EEG findings for those that succeeded and those that didn't, right? I mean, you need a comparator usually when you make those assessments. I don't think it's been valid. Yeah.
In our paper, we look at the EEG changes during the surgery, which is about maybe in average 1 week after their trial period. So pre-op period is 1 week. I think it is pretty close to the trial period, you can say. And EEG can be recorded at different periods during the treatment, and you can compare the changes. But there are different factors like different programs, a different location in the electrode. You have to control a lot of parameters to be clearly, correlating the changes to the single SCS program, I guess.
Okay. All right. So, Lily, will people be able to get these slides? These slides will be available afterwards, right?
Yes. Yes. This recording is going to be available online before the end of this week.
Okay. There was a question about, is there any effect that people saw in the literature of SCS on symptoms of depression or anxiety? I mean, I don't know that I haven't seen anything directly. I assume if a person gets relief, it will improve their mood and their anxiety. As far as the pain areas of the brain being related to emotion, it's most related to reward and analgesia and the dopamine system. So I can tell you that prefrontal cortex is dopamine related and has its own dopamine projection. The insula receives reward related and analgesia related dopamine from the A10 ventral tegmental nucleus in the same trajectory as the nucleus accumbens, which has been studied so much for addiction. So there are monoamines that exist in these areas, but I don't know that I've seen anything where spinal cord stimulation changes the monoamine levels or affects anxiety or depression. Dr. Ferguson, someone's asking about location and actual settings for the abdominal and pelvic pain with the subperception settings.
So I'm sorry, can you repeat the question?
Where would you put the leads and what would the settings be to capture abdominal or pelvic pain using subperception?
So a little bit of a preamble. So before I would do a spinal cord stimulator trial, I would probably do a splanchnic nerve block for abdominal pain or a superior hypergastric plexus plane. Assuming that works, then I would put the leads at the top of T6 to start with for abdominal pain and probably the top of T11 for pelvic pain. And the settings tend to vary between patients, but I listed them in the slides. But really, I just have the rep find where that unique patient, where their perception is, and then I just turn it down and then run fast. But I don't do paresthesia mapping because it's very, very difficult to get paresthesia overlap with the abdominal or pelvic pain because it's not somatotopically organized. It's very, it just doesn't work that way. So I just run fast, and if they get good relief, then that's where I leave them. By good, I mean like 80 to 100%. If they don't get that amount of pain relief, then I just adjust the leads level by level until I get the amount of pain relief they're looking for. But the fastest, it's really, really fast. So it's like, turn it on, see how they do.
Okay, I think they were just looking for some practical numbers there. Did anyone find anything in their work researching this relating to sex as a variable? Male or female sex as a variable for spinal cord stimulation mechanisms? I mean, theoretically, I can only imagine the effect of the hormone on immune responses. We know that exists. But did anyone see any significant work on male or female differences in spinal cord stimulation patterns? No. Yeah, I didn't run across any either myself. There's a question on a good review on the effects of diabetes as it relates to the lamina layers, I guess the differentiation issue that I brought up. There is a rat model of diabetic neuropathy called streptozicin diabetic rats. So there might be basic research on that topic. I did find a review article on painful versus painless diabetic neuropathy, looking at what might account for the differences between the painful and the painful diabetic neuropathy. I can see if I can find that article again and post it or give it to Lily. Let's see. Does anyone want to comment on the future of closed loop SCS? What the role for that will be, or what particular benefits it might be in the future?
I guess it's on my talk for the Ecap, and just it's basically what's going to do. We're probably going to allow a little bit less battery consumption as well, because it's going to be in real-time adjustments. So it's not going to be a definite set for the neuromodulation. However, to my knowledge, it's not still introduced in the US. It's been very good success in Europe and Australia. So we're hoping that it's going to be available here soon. But that's the idea. It's just, you know, we don't, every single time you use neuromodulation, you have to have a specific dosing for the spine. And as I mentioned, there is a correlation to the amount of CSF, position of the electrodes, how much intensity, and so forth and so forth. So being able to do it in real-time adjustment, you know, beat by beat, basically, impulse by impulse, it's essential to kind of be smooth and without ups and downs, and, you know, much less battery consumption, hopefully, in the long run.
Yeah, and one mechanism for spinal cord stimulation tolerance is if we could be inducing, I suppose we could be inducing cell death in those GABAergic neurons if we're pushing them all the time with our energy, and we're causing the inhibitory neurons to fire. We could be causing that could be an issue with development of tolerance over time, is if we're inducing even cell death into the GABAergic interneuron population. So the lower energy we can use and the less hard we push the system, probably the better for our success over time. Dr. Telkis, there's a question for you about, will TDCS for the hotspots T3, 4, or F4 ever be in alpha help reduce pain sensation? So they're talking about transcranial, I guess, DCS to T3, 4, F3, 4, those sites which light up. I think they're asking if stimulation, transcranial stimulation to those points would be useful in blocking nociception or pain.
I'm not an expert on that literature, but based on the EEG literature, I can say that possibly. Yes, because those are the regions that EEG studies also reported activation in changes in alpha and theta bands. So as long as you can induce changes corresponding to pain relief, why not? But I think maybe I can add to the previous question in terms of the closed loop, which is also correlated to biomarkers. I want to emphasize that before, I think the first step before the closed loop system, developing closed loop system, is to characterize objective metrics to assess the SCS, different waveforms, and assess their effect in depth. So it's going to be independent from, you know, patient-reported outcomes. So we can then develop closed loop systems based on those biomarkers. And I guess you can also, as long as you can modulate those neural networks in favor of pain relief, I guess it doesn't matter which modality you are using. Non-invasive and cheap modalities, of course, we preferred.
Okay, and I think we'll wrap up with a pair of related questions. There was a question about how do we select patients for neuromodulation, and does anyone see a role for neuromodulation in acute pain? So how do we, I think, how do we select patients for spinal cord stimulation and/or choose one of these waveforms for the different types of syndromes that we see? And someone's asking if you know, the benefits of spinal cord stimulation should also be there for acute pain. Is this something we should be studying more? Is there a gap in our practices for the utilization of spinal cord stimulation in acute pain? I think, you know, for acute pain, it would be tough to for pain of less than 3 months, it'd be tough to justify an invasive procedure like spinal cord stimulation to go to trial to implant in something that may resolve itself. And so my answer to the acute pain question would be to rely on probably nerve blocks or even sympathetic blocks to try to reduce neuropathic pain initially and see if it doesn't improve and fail to become chronic. So I don't personally see a role for implanted spinal cord stimulator systems for acute pain, given that they're permanent systems. I'm not aware of any temporary systems that might be of use, although we do have temporary 60-day peripheral nerve stimulation systems available. But I don't see spinal cord stimulation being that valuable for acute pain going forward. But others can speak to that and address maybe that issue of how you select or how you decide who you're going to trial for spinal cord stimulation and what the thought process may be for selecting one of the different systems or waveforms. Either Dr. Ferguson or Anna Tescu can go first. How generally they select patients and choose.
I'll jump in on how I select patients then. So I think there's a careful, what everyone does, informed consent, but I work really closely with a psychologist to appropriately screen and vet patients. So I can say that my psych approval rate is not even close to like 100%. It's probably more like 50%. Like they go through a full pain coping, pain pacing regimen, and at least there is data to show that if the less opioid someone's on, the better they're likely to do. If they have any underlying psychological issues like depression, et cetera, that's treated. And then per the NAC guidelines, I have them not smoke. Those are, I'd say, the largest things: not smoking, minimize or off opioids, and treat underlying psychological disorders. If they respond to, if they have all the standard, if they have the standard conditions like complex regional pain syndrome or post-laminectomy syndrome, it's kind of straightforward. If it's another neuropathic condition, consider like splanchnic nerve blocks, celiac plexus blocks, suprahypergastric blocks, etc. If patients have head to toe pain, like everything hurts, odds of them doing well are not great.
Yeah, so essentially sensitized persons not necessarily going to do well. We didn't talk about DRG stimulation, but that's another option for focal or regional neuropathic pain syndromes. And a lot of people do trials where they swap in the middle of the trial. So they'll try one system with 10,000 hertz, and then they'll, after a couple of days, they'll switch to fast or to a burst program or something. And what I'm told is that 75% of the time it doesn't make much of a difference, but maybe 25% of the time it does. And but I think a lot of us just do it empirically, we try to certainly for, I think, axial conditions, you're going to stay away from, you're going to go with non-paresthesia, you're going to use HF10 or burst or fast or one of those. Dr. Anadescu, do you have anything to say to that?
No, it's just, it's a good, I mean, we always do the spinal cord stimulation, the psychological evaluation as well. This is not the first one, the first treatment we actually introduced to the patients. We try different things because many patients do respond to less invasive treatments. However, at some point in time, if we think the patient has neuropathic pain and doesn't really stop the response to other treatments, we go ahead and do. In terms of which company we use, as you mentioned, they're pretty much interchangeable. It's just, so we had more success in some patients with some companies, some success with others. It's just, it depends of coverage. It depends of how wide, for example, if we want to cover 2 or 3 vertebral body, we may be using 16 contacts as opposed to 8 contacts. But again, it's just case by case, basically, evaluation.
Yeah, and I think that there's a limitation here. When you're dealing, I mean, there's difficulties with doing research on interventional or invasive therapies anyway, with regard to blinding and whatnot. But also it's expensive, and a lot of this is industry-sponsored research. So industry is not, they're not interested in showing patients in whom it may not work, or in going head-to-head in a situation where their product may not be shown to be superior, it may even be shown to be inferior. So generally the research is done in such a way as to show that it is effective compared to usual medical management or to paresthesia-based older waveforms. But to really try to drill down and determine what patient factors are predictive of success versus not of success, and to compare waveforms head to head and whatnot. I mean, that would have to be done outside of the type of industry trials that are currently sponsored and paid for by industry. It would have to be something that was publicly funded, or it would have to be something research that came out of, you know, retrospective databases and whatnot with carefully collected information and stuff, because I don't think there's going to be industry sponsored research to answer some of these questions. It's just not going to happen. Okay. I think that'll probably do it. I'll check quickly to see if any more of the questions came in, but I don't think, I think we're good there. So thank you everyone for joining us. It's past 6:30 now. It's almost 6:45. Dr. Telkis, Anatescu, Ferguson, thanks for all the work you did putting this together.
Thank you.
This will be up. This will be posted online by the end of the week. And if anyone on the call was interested in any of the references or articles or whatnot that were cited in the talk, once we get a look at the slides, we should be able to provide the papers as well to back up the information. The slides provide a little more detail. Okay, Lily, are we good?
Yes, thank you so much,
everyone.
Thank you. Bye.
Thank you.
Night.
Bye.